Hello everyone. My name is Dr. Monica from NIET Pharmacy Institute, affiliated to APJ Abdul Kalam University. I am taking the subject BP303T Pharmaceutical Microbiology. In my last lecture, I have covered up unit number 1 that was related to your introduction part, introduction to microbiology, isolation. We studied about quantitative estimation of various microbes. We studied about various microscopes which can be used for microbes analysis. So, in this after covering unit number 1, I have started with the unit number 2, and in unit number 2 we have started up with the identification part. And in identification part, if you remember, I already covered these topics, that is direct examination. We can see microorganisms using various microscopes. Now, using various microscopes, we can do the direct examination as well as we can go for the staining techniques. Under direct examination, we covered up total wet mount method and hanging drop method, by which we can analyze the motility of our microorganism, we can see the structure of microorganisms, we can see the shape of microorganisms. And by staining techniques, we covered up, in staining techniques we covered up the simple staining and differential staining. In simple staining, I told you that we can use only one stain and we can analyze the structure of microorganism, whereas by using differential staining technique, in this we can use more than one stain, 1 to 2 stains. Like in gram staining we used crystal violet, safranin, and in acid fast staining I told you you need to use carbophol fuchsin and methylene blue. So, in this way, using more than one stain, we can differentiate the characteristics, we can differentiate between the gram positive and gram negative bacteria or acid fast or non-acid fast bacteria. So, these all topics we have covered in the lecture I have taken before. In today's lectures, I will mainly focus on the biochemical test. Now, what we mean by biochemical test, and in this also we will mainly focus on the IMViC test. Now, what actually the biochemical tests are? Biochemical test mainly, the biochemical word indicates that chemicals released by the bioorganisms, means some types of enzymes, some types of chemicals are always released by the bacteria, by the fungi, by the viruses. Different type of microorganism release different kind of chemicals, and analyzing those chemicals with the help of identification techniques, that what we call as biochemical test. Biochemical tests are the tests used for identification of bacterial species based on differences between their biochemical activities, means their biochemical activities differ, means they release different kind of enzymes, different kind of chemicals within the preparation, which that chemicals can be analyzed with the help of various analytical tools or various analysis staining techniques, various techniques. We can analyze those enzymes, we can analyze those chemicals. These tests mainly depend upon the presence of certain enzymes, like some of the bacteria always release catalase enzymes, some of the bacteria may release tryptophanase enzymes. So, depending some of the bacteria may release oxidase or urease or gelatinase enzymes produced by various kinds of bacteria. So, different kind of bacteria release different kind of enzymes, which those enzymes can be analyzed because these enzymes may degrade, may metabolize many products. So, we will, in biochemical test, we will analyze that whether those bacteria are releasing such kind of enzymes or not. Those bacteria will be identified by using biochemical test, and in this also mainly we have to focus on the IMViC. Now, what IMViC stands for? IMViC, as the name indicate, I stands for Indole Test, M stands for Methyl rate test, V Vogues Praskauer test, and C is your Citrate Utilization test. So, I stands for Indole, methyl, M indicates methyl red, V indicate Vogues Praskauer, and C indicates Citrate Utilization test. So, in this test we mainly focus on these, that is IMViC test, in which we will try to find it out the microorganisms which are releasing citrate enzyme, which are releasing tryptophanase enzymes, which are releasing which release acids at the end product, which release acetone as an end product. So, those chemicals, those enzymes can be identified by using these type of biochemical tests. So, one by one we will discuss all these tests, what we need to do to analyze such kind of bacteria, to identify such kind of bacteria. So, if we see go for Indole test, what we are going to do within the Indole test, that is the main principle of this test is that this test will identify the bacteria which actually release an enzyme known as tryptophanase. The bacteria which release tryptophanase enzyme will be identified using this technique of Indole test. Now, how we are going to identify? What you need to do is you need to prepare a broth which will contain tryptophan. Means you in the lab what you can do, you can prepare a special type of a broth which will contain tryptophan, and after that whatever microorganisms we need to identify, we have to inoculate those microorganisms, those bacteria within that broth. The broth containing tryptophan will be inoculated with the help the microorganisms, with the bacteria which we need to identify. And once you have inoculated, after that you have to incubate those petri plates containing tryptophan broth along with the microorganism, along with the bacteria which you need to identify. And once you have incubated, what will happen? When you will incubate, the tryptophanase enzyme will be released by the bacteria. Now, tryptophan, the broth was containing tryptophan, and as tryptophan was there within the broth, and bacteria which you have inoculated, those bacteria are releasing tryptophanase enzyme. Now, that tryptophanase enzyme will do? It will degrade tryptophan and it will metabolize tryptophan into indole and pyruvic acid and ammonia. 3 byproducts will be released once the tryptophan will be metabolized with the help of enzyme which has been released by the bacteria, that is tryptophanase enzyme, that will degrade the tryptophan broth, and tryptophan will get metabolized into 3 byproducts like indole, pyruvic acid and ammonia. And that indole which has been released from that reaction, that indole will be identified with the help of the reagent named Vogues reagent. What we need to do? We have to add Vogues reagent into that broth which has metabolized the tryptophan due to the release of tryptophanase enzyme. The indole which has been released, that indole will be identified with the help of a reagent named Vogues reagent. When you will add Vogues reagent to that broth, what will happen? It will convert the broth into red color. The red color will generate as you are going to add Vogues reagent. When you will add Vogues reagent into the broth which has been metabolized with the help of tryptophan enzyme, tryptophanase, so the indole which has been generated, that indole will be identified with the help of a reagent named Vogues reagent, that will convert the broth into red color, and the red color is the indication that your bacteria were releasing tryptophanase enzyme, and those species are the those bacteria are the bacteria which are releasing tryptophanase enzyme. So, this is a technique by which we have identified the bacteria which release tryptophanase enzyme in the preparation. Secondly, we can go for methyl red Vogues Praskauer test, and what we need to do in methyl red Vogues Praskauer test, methyl red is these both are the reagents, methyl red as well as Vogues Praskauer, both are the reagents which you need to add into the preparation, in which you need to identify some of the bacteria. Now, which type of bacteria we actually identify using methyl red Vogues Praskauer test? The bacteria which release acid as the end product. When they ferment, that is glucose, when they go for fermentation of the glucose, what they will do? They will make the preparation acidic, and the preparation will be identified with the help of methyl red, and some of the bacteria also make the preparation basic in nature. So we can differentiate between the bacteria which are releasing acids at the as an end product, and we can identify the bacteria which are making the preparation basic by the release of an ingredient known as acetone. So both these can be identified using methyl red Vogues Praskauer test. Methyl red test, commonly known as MR test, we also called it as MR test, that is methyl red. Methyl red is a reagent which we need to add into the preparation containing some of the bacteria. Now when you will add that reagent of methyl red, what it is going to do? First you need to know whether your organism, when you have inoculated your bacteria within the media, you have some media, when you will add, that is what we call as bacteria to be analyzed, we will add those bacteria in that product containing glucose. What the microorganisms are going to do? They will ferment the glucose, and that fermented glucose will get converted into that is acid product. They will release at the end, that is lactic acid, acetic acid, or various kinds of, they will create the environment which will be acidic in nature. And when the environment is acidic, that acidic environment will be analyzed with the help of a indicator known as methyl red. Methyl red is an indicator which is red at pH 4.4 and which is yellow at pH 6.2. So when methyl red will be added to the preparation containing bacteria containing bacteria which are metabolizing glucose or fermenting glucose and releasing some of the end products which are acidic in nature, those acidic environment will be that is, we will identify that acidic environment with the help of reagent name as methyl red. When you will add methyl red indicator into the preparation, what will happen? If red color will be formed, if it generates a red color, that indicates that your preparation is becoming there has been created an acidic environment within the preparation. So acidic environment will be identified with the help of a reagent known as methyl red, which shows red color at pH, acidic pH, and which shows yellow color at the basic pH. So we will be able to differentiate whether our bacteria are converting our glucose or fermenting our glucose and making the environment acidic. So acidic environment will be that is analyzed with the help of methyl red. If the organism produces large number of organic acids, now a large number of organic acids like formic acid, acetic acid, lactic acid, or succinic acid, then those acidic environment after fermentation will be analyzed with the help of methyl red. But what will happen in case of MR negative microorganism, means the bacteria which are giving negative results for methyl red. If the preparation will not convert into the red color, or it remains yellow in nature, or it will remains in the color which it already had. Now what will happen? That once you have, in case of MR negative, means if the preparation is not becoming red, it remains yellow in color. In that case, what will happen? That glucose will further be metabolized, that glucose will further be fermented. So MR negative organism will further metabolize the initial fermented products into various acetones. So when the acetonic environment will be created with the by bacteria, that if acetone is released as an end product, that will give the MR negative test. Means what you have to understand, that MR positive and MR negative means methyl red positive are the bacteria which make the environment acidic, and MR negative bacteria are the bacteria which will make the environment basic in nature. They will ferment the glucose and convert it into an acetone product, whereas MR positive bacteria will make an environment acidic and they will ferment the glucose and will release acetic acid, succinic acid, acids in the environment, and they will give red color at the last. And if you obtain yellow color, that means your preparation is MR negative. So as you can see even in this picture, that MR positive are the bacteria which are converting the preparation into red, and the preparation, if the preparation you see at the end is yellow in color, that means they are MR negative, means they are not making the environment acidic, they are fermenting the glucose into different acetonic products. So, MR positive will be indicated by red color and MR negative will be indicated by yellow color. Along with that, I will like to tell you that the microorganism, the bacteria which gives MR positive results and they will give VP negative results. If MR results are positive, then VP result will be negative. But if MR results are negative, then VP results will be positive. Means the bacteria MR will give you that those will identify those bacteria which will make the environment acidic, but Vogues Praskauer is the reagent which will identify the microorganism, which will identify the bacteria which make the environment basic, means they are releasing acetone as the end product. MR negative bacteria always show VP positive results and MR positive bacteria always show VP negative results. So, in this way these both the tests you have to carry out that is simultaneously. MR test and VP test are always carried out simultaneously to identify, to even cross check whether MR positive microbacteria are there or not, or MR positive will give VP negative test and MR negative bacteria will give the VP positive test. So, now what actually Vogues Praskauer reagent is? Vogues Praskauer test, commonly known as VP test, is used to determine the ability of some of the organisms, some of the bacteria which mainly make the environment basic, which will release or they will make the environment neutral or they will make the environment basic, and at the end of that is glucose fermentation, what they are going to release as an end product, they will release acetone as an end product, and that acetone will be analyzed with the help of VP reagent. And that VP reagent, what color it when we will add that Vogues Praskauer reagent into the preparation in which we have inoculated bacteria and they are making the and they are fermenting the glucose and making the environment acetonic, or they are making they are converting from acidic environment to basic environment, and at the end what they will do, they will show a pinkish red color at basic pH, where Vogues Praskauer will show pinkish red color. When you will add Vogues Praskauer reagent, you will observe pinkish red color at the end, and VP positive test means pinkish red color and VP negative test means that your preparation remains in the natural color. It will have the same color what it was having before. So, pinkish red color indicates VP positive microorganism, that means those bacteria are releasing acetone as an end product. So, the microorganisms which make acidic which makes the environment acidic are that is analyzed by methyl red, MR will be positive test, and the bacteria which make the environment which will release acetone as the end product will be analyzed with the help of Vogues Praskauer reagent, and Vogues Praskauer test will be positive and will be analyzed by the pinkish red appearance of the broth, and that pinkish red broth will let you know that your preparation contains bacteria which are fermenting the glucose and they are releasing as an end product, that is acetone. So, as you even can see in this, if you have reddish pink color, that means VP positive, and if the preparation remains yellow in color, that means VP negative. But that you have to remember that if the test is VP positive, means MR test will be negative, and if MR test is positive, then VP test will remain negative. So, this is what we call as methyl red Vogues Praskauer test. By Indole test, we analyzed the bacteria which release tryptophanase, and that was indicated by the indole, and that indole was analyzed using the reagent known as Vogues reagent, and by methyl red Vogues Praskauer test, we have analyzed the bacteria which are either fermenting the glucose and make the environment acidic. by methyl red, and the Vogues Praskauer is letting us know that those analyzing those bacteria which are making the environment acetonic, means they are releasing acetone as an end product. So, these are these two tests are related to your biochemical test. One is Indole test and second one is Vogues Praskauer, that is methyl red Vogues Praskauer test. After that comes the citrate utilization test. Now, what we mean by citrate utilization test? As you remember in my last slide, in my starting slide, that is Indole test. Indole test is indole production test, means indole is generating, indole is released as a product, as a byproduct, as a metabolic product from the tryptophan. Tryptophan was metabolized and then indole was produced, means that is why we all use the word indole production for this indole test. But for citrate test, what we are using the term, that is citrate utilization. Now, what we mean by citrate utilization means we have to take a broth which must contain citrate. Citrate will be metabolized here. In the first test, indole was produced, but in this case citrate is going to be utilized, is going to be metabolized by the enzymes, whatever the enzymes are being released by the bacteria. Now, which will be the enzyme which will metabolize citrate? The enzyme will be citrase. This test will identify the bacteria which will release the enzyme known as citrase, and citrase enzyme, the bacteria which release citrase enzyme, and if we are having the broth which contains citrate, thus those citrase enzyme will metabolize that citrate product within the that is broth. So, citrate agar is actually used as a media for such type of for the analysis of such type of bacteria. So, citrate agar is used to test the ability of an organism which utilize citrase. They will utilize citrase because they will metabolize citrase into the byproducts like oxaloacetic acid or acetates. Now, what will happen? Citrate will act as a source of energy for such kind of bacteria. When you will add citrate into the broth, citrate will act as a source of energy for such kind of bacteria. The agar medium containing citrate as a sole carbon source, it will act as a carbon source, it will act as a nutrient for the growth of bacteria, and as the bacteria will grow, they will release an enzyme known as citrase, and that enzyme will break down this citrase enzyme. The enzyme released by the bacteria known as citrase enzyme will break down the citrate into oxaloacetic acid and acetates. Whereas, when oxaloacetic acid will be released or oxaloacetate will be released, that oxaloacetate will further be metabolized with the help of those enzymes into pyruvate and carbon dioxide, means they will release pyruvate and carbon dioxide at the end. You can see this reaction, how the reaction is being going on. What will happen? Citrate, we have taken the broth that was containing citrate. So, that was containing citrate, and when you have added, when you will inoculate the bacteria which are releasing citrase enzyme, what will happen? That citrate, as your bacteria will release citrase enzyme, what will happen? That citrate will be metabolized into oxaloacetic acid, and that oxaloacetic acid will further be metabolized into pyruvic acid and carbon dioxide. So, the release of carbon dioxide induces the metabolism of ammonia salts. Now, what will happen? Excess amount of sodium will remain within the broth. That sodium will be consumed by the carbon dioxide. They will make the environment now basic. When sodium and carbon dioxide will combine, what will happen? They will release sodium bicarbonate, and once sodium carbonate will be formed, and sodium carbonate will be analyzed with the help of various kinds of indicators which can differentiate between acids and base. Like the reagent which we are going to use within this is bromothymol. Bromothymol blue is the reagent which we will add within the preparation to analyze whether our the bacteria has released citrate enzyme or not, that will be analyzed with the help of bromothiamol indicator. Now, bromothiamol indicator is an indicator which shows different color in acidic environment and which will show different color in basic environment. Mainly it has green color in acidic environment and it has blue color in basic environment. In acidic environment it will have green color and in basic environment it will have blue color. And when your citrate will be metabolized into oxaloacetic acid and then the color of the broth will remain green in color. So, after the color will remain green and it will that will metabolize into pyruvic acid, and pyruvic acid will release along with carbon dioxide. Carbon dioxide will combine with sodium and it will make the environment basic. Sodium carbonate will be formed and your environment will become basic in nature. So, that basicity of the media will be analyzed with the help of bromothiamol blue indicator. Your preparation will turn from that is green color to blue color. As you can see in this, that when citrate as you can see this, here we have blue color and here we have green color. In the starting your preparation in the acidic environment, when there was oxaloacetic acid release or pyruvic acid were released, your preparation will be green in color. But when carbon dioxide will combine with sodium and they will make the environment basic, then it will become blue in color. So, this is what we call as citrate utilization. So, in this whole lecture what we have studied, that is first we have studied that is mainly we have focused on the biochemical test. In biochemical test we have covered up IMViC test, and in IMViC test I first covered Indole utilization test, Indole production test. Second I covered up that is methyl red Vogues Praskauer test, and in the last we have covered up citrate utilization test. So, in biochemical test, in Indole test, what we try to analyze? We try to identify the bacteria which are mainly releasing an enzyme known as tryptophanase by using a broth which was containing tryptophan. And after that what we have done? We have used methyl red Vogues Praskauer test to analyze whether our bacteria are fermenting the glucose and creating an acidic environment, or our bacteria are fermenting the glucose and making an environment that is acetonic in nature, or that is towards neutral pH, moving towards neutral pH or basic pH. So, that was analyzed using Vogues Praskauer reagents. And after that I have covered up the citrate utilization test, and in citrate utilization test we tried to find it out the bacteria, we tried to identify the bacteria which releases the enzyme known as citrase enzyme, and that citrase enzyme is being analyzed using an indicator known as bromothiamol. Bromothiamol as an indicator which which differentiate between acidic environment and basic environment. At basic environment it will become blue and it shows that citrate test is positive. So, this was all about that is biochemical test. So, these are the references which you can follow, and for that is practical point of view I always mention Chiaraneja to be followed, and for theoretical point of view you can go for Pelzar or Prescott. So, this was all about your biochemical test, and here we are winding up that is identification of bacteria which was containing simple test, which was containing staining, which was containing direct examination, which was containing biochemical analysis. So, thank you so much.